Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves.
Welcome back to Careers in Discovery. Today I'm joined by Mikko Turunen, founder of RNatives. Welcome, Mikko.
Thank you, and thank you for having me.
I'm fascinated to get into your story. I suppose a great place to start is to talk a little bit about the work that you are doing with RNatives at the moment.
Okay, RNatives is a 4-year-old company doing RNA activation, which is not very widely known phenomenon, but it's like the other side of the RNA interference, but it's a nuclear small RNAs that regulate gene expression at the level of transcription. So they target non-coding transcripts in gene promoters, and that these RNA binding in gene regulatory areas leads to epigenetic changes in histone tails and can either activate or downregulate the targeted gene depending on which location it is targeted. And we are utilizing this phenomenon for therapeutic purposes. Our lead asset, which is almost ready to clinic, regulates gene called VEGF-A. It grows new blood vessels, so treatment of hypoxic diseases such as myocardial infarction or peripheral artery disease can be treated with this, and we have great data from animal experiments already. But then again, we are a platform company, and we have asset already identified and confirmed in wet lab for Parkinson's disease. And other indications that we are looking is wet AMD and some oncological projects together with our partners in other companies. And we are really agnostic, basically all the genes that are epigenetically regulatable and have long non-coding RNA, the promoter, are potential targets for our technology. And we use different formulations for the delivery. It can be either as a naked RNA or as a RNA in exons, which are extracellular vesicles, or we can use LNPs as well. And we were actually first ones back in 2009 to show that RNA activation can be used in animal models. That was a peripheral artery disease model, or mouse hind limb ischemia to be more precise, and where we activated VEGF-A and we restored the blood flow to these mice. And that was a circulation research paper back in 2009. So what I do in RNatives, as a founder, I'm involved basically in everything. We are still a small company, but we are now starting to grow, and we are having exciting times now that we just are closing the new financing round, and we are in the process of recruiting a new CEO for the company. So we now can make the US market entry finally. It has been quite challenging last 2 years, because of the financing market, but I'm now confident that we are moving happily forward.
Congratulations, it's a huge achievement. From a groundbreaking discovery to overcome the hurdles that you have, that's certainly something that I'd like to dig into a little bit later on in our conversation.
Certainly.
Suppose I'd like to take you back to the very beginning, to your early days, and what it was about science that piqued your interest, particularly to get into the areas of drug discovery.
As a kid, I was always very curious about science. I read a lot of popular science journals and stuff like that, and I was very curious about new technologies and everything. And in the early 90s, when I started applied to the university, the biotechnology and molecular biology were really in a transition phase, let's say. So all the new tools have been developing, PCRs were there, sequencing, and so I found it fascinating, and that's why I went to former University of Kuopio. It's now University of Eastern Finland. I went to study my master's thesis, and I just got pulled by the science so heavily that it took me 3.5 years to get my master's thesis.
Wow.
And I immediately continued 97 in Professor Seppo Yläherttola's group as a PhD student. And again, 3.5 years, I got my PhD in 2000. And so that's how things got started. I just was in a vacuum of science. It just overtook me, basically. And I always thought that we really don't know. We only have scratched the surface of the knowledge. So there's so much still to learn. The world is not ready. And that was my attitude. During my PhD, I did mostly in vivo studies. Not that much hardcore molecular biology, but at 2001, I went to do my postdoctoral studies in Luigi Naldini's group in Torino, and he was one of the original developers of lentiviral vectors, which we saw very important at that time to learn that technology. Yeah. And we certainly did. When I went there, whole family moved there. I had two small children already.
Wow, and, that's a big risk to take.
Well, yeah, that was very, very big risk to take, and of course it had its cost, because you are, as a postdoc in abroad, you are expected to work a lot. So I did something like 70 hours weeks or so.
Wow.
So that had an effect on the family life, of course. And actually, eventually, when we got back from my postdoc back in 2003, we decided to divorce. So it had a sort of personal.
Yeah,
but it's things happen, in life, and I was just so driven by science. But anyways. I've still been able to be a good dad for my two daughters, and things just sometimes go south, and you just can't affect them. But anyways, so after when I came back to, I want to mention that in Naldini's group, I was really given hardcore molecular biology task to do, or project. So I was asked to do gene expression profiling with Affymetrix microchips, which were a new thing back at that day, to study the recipient cell response to lentiviral vectors.
Okay.
And that was a big challenging study, because when we did, and it was expensive, and when we got our first results, we were very surprised, because we didn't see much transcriptional response, and there must be an error. So we did 2 more experiments, always adding more and more controls there, and finally we believed, okay, cells don't really much respond to the lentiviral of that. But sometimes you just have to trust the data. It is what it is, and not try to reformulate your hypothesis after the experiment has been done. But anyway, so I got back 2003, end of 2003, and spent a couple of years as an academic postdoc in University of Kuopio. And then I joined 2005 to Therapeutics, which was same Professor Seppo Yläherttola, his company, and it was it was listed in London Stock Exchange, and it had a subsidiary here in Kuopio. So I spent 7 years in that company, and that was really beneficial concerning the current me running this company. So I learned really valuable lessons from those years, both positive and negative. 2012, that company failed in its clinical trial, and it was ended, delisted. And at that time, I was able to get my own project funding from Academy of Finland. And during that time, the plan was to use the first year as a senior sabbatical in San Diego.
Okay.
And so I moved to San Diego, and and there was Kevin Morris's group in the Scripps Research Institute. And I already knew Kevin. We had invited him to Kuopio earlier on, and were really like buddies. So it was a really good place, because Kevin was actually first one to show back in 2004 in his science paper that small RNAs can silence genes in transcriptional level. So he was the pioneer in that. And the Longcheng Li was one that showed back in 2006 that small RNAs can also activate gene transcription, depending on if you target the activating site into gene promote. So so I want to mention that during 2007, I met my current wife, who came to do her master's thesis for me, and we've been working 17 years together with this RNA activation, which is the foundation of this company. And it's if you will, it's like a family business. I would say we live and breathe 24/7, 365 this company. So it's like our common baby, if you will.
I love that idea.
Yeah. So, but I think that's the reason that we have this resilience, and we were able to pull last 2 very difficult years together. We are both all in on this, and we know that we are correct about the technology. Yeah. So we have very strong view of this. So So I mentioned that we started doing that study in 2007, and then when I moved to San Diego 2012, the data was so good from the animal experiments, and the other experiments started to make us wonder that maybe this is a natural phenomenon. And actually, when we got started, we used lentiviral vectors with hairpin RNAs, which are structurally very similar to microRNAs. And they were very efficient both in the treatment of hind limb ischemia and also in animal models in myocardial infarction. And during my time in Scripps and discussing with brilliant postdocs there from various parts of the world and all the other people, I started to really think, there must be nuclear microRNAs that do this thing naturally. We must have been accidentally mimicking something that exists in nature. And we started doing the bioinformatic analysis from the VGFA promoter, which is our model gene in this case. So we found that, MIR466 actually heavily targets VGFA promoter, and especially this important loci, which is a transcription factor binding sites for ATF4 and others, and CREP2 and all of those. So we really started concentrating our focus on MIR466. So when I came back from San Diego 2014, and I must say that was quite difficult to return, let's say bittersweet, because Tia had been here in Kuopio all the time, and we weren't yet married. We married 2015, but so it was a long distance relationship, and I had time of my life in San Diego. I lived 2 blocks from the Pacific Beach, and
wonderful.
I had great friends there, and so it was quite different. So it took 2 years for me to lose my tan, for example. I swam in the sea, I learned surfing, and I drove a red Mustang, and life was good.
Dream.
I came back to Finland in September 2014. It was raining horizontally, and it was pitch dark, and it's like, why I returned? But
why am I here?
But it turned out good. So year after my return, we got married with Tia, and continued this work with my academic project. And the data during that time strongly convinced us that we really must start a company around this technology, because we really now understand something that is not widely known. So I got Business Finland funding, research to business funding for 2 years, but back in 2017, if I remember correctly. And during that time, we started planning the spin-off and the technology transfer and all of that for the new company. And we got funding during 2020 for company, and it was agreed with our seed funders that we will form a Delaware company, and that will do the technology transfer with the University of Eastern Finland, because then we wouldn't have to do very expensive Delaware flip later on. Right. once we grow. And we got commitment for the seed funding, and what happened? COVID. And so I was like, I can't travel to US. How can I open a bank account? US banking system is pretty conservative, at least if you compare to Nordics. So it was a lot of more than 100 phone calls here and there, and pulling all the connections and tricks I could do. And eventually from US embassy in Helsinki, there was a business development person who knew the Citibank boss from Europe. And then I had put me in connection with her, and then I pitched the whole thing on the phone for her and say, okay, we really need the US bank account. And okay, I will talk to my colleague in New York. And then her colleague calls me like 4:00 in the morning because of the time difference, and I'm just like really blurry, but I was able to pitch the whole story during that nighttime call, and he agreed that okay, this is legitimate and we can open a bank account. But then there was still it was a lot of work, but still somebody had to physically go to the bank to get to sign all the keys off and everything like that. But luckily, our law firm in Finland is Borenius, which is the largest and best law firm, and so they had a side office in New York. So their paralegal was able to take care of that. And after 6 months, we finally got the whole thing sorted out.
That's a lot of navigating, isn't it?
Yeah, it really took quite a lot of effort, but it really pays off because our also strategy has always been to try to partner and work with the best companies. So now that we were ready to pay maybe the higher price of Borenius compared to others, it really paid back because they had those resources which would be utilized. So we got started with the company, and then we founded the Finnish subsidiary, which is completely owned by the US entity, and we have to build a laboratory. So
where do you start?
It's in Novapolis here in Kuopio. There are a lot of startups there. So they built the rooms for us, but we had to find all the equipment, of course, and all the hoods and biolaminas and all of that. And of course, being a startup, we didn't have millions to invest in the laboratory. So what we did, we realized actually that there's a lot of hospitals that are being renovated in Finland, and there's lots of auctioning those, because they always have the laboratories there as well. So we got dirt cheap, all the everything basically for the lab. So, and we had like a chemical hood, which is just like new one. We got it for free. Biolamina, which can cost somewhere 15,000 to 20,000 if you buy it new. We bought it from auction in Jyväskylä for 300 euros. Wow. Yeah, and all of this very, very cheap. Some facilities and machines we bought as new, like microscopes and centrifuges and all of that. Yeah. But in the end, we were able to save hundreds and hundreds of thousands just by doing the work. And we had this startup spirit, so what we did is that always when we won the auction, so I put everybody in the van and we drove to the hospital there, and we have the drills and screwdrivers and everything, and just pack it and push it and organize it into the car.
And you have to roll your sleeves out when you're in a startup. You need to roll your sleeves up. You need to get in the roots and get dirty, don't you?
Yeah, that's exactly. I really felt I was doing like mechanical work for some time, all of that. But that way we were able to save enough money so that we can do the required animal studies with CRO.
Yeah.
So it's always that kind of struggle business, at least in European startup. I know in US, at least back in the day, the seed funds could be in such a big numbers that you can, just buy, say, Thermo Fisher, okay, build the lab and just pay the bill, but not in Europe, I think. So
every penny counts, doesn't it? And what I think is really prevalent in your story is where there's a will, there's a way.
That's sort of the theme. Yeah. And then when we got the lab, we started doing data and everything was going really well. And we were able to start negotiation with a VC in London. And eventually, autumn 2022, we got the term sheet for 24 million pounds for Series A. And okay, now we are heading to clinic finally. And okay, and now things start really scaling. Okay, what then happened is that first of all, war started in Europe, and then the financial crisis. So during the negotiation, when we were negotiating the term sheet, the Silicon Valley Bank went down, and that affected the VC in London, and their LPs up from them withdrew, so that paper just vanished in the air. So early 23, we were in a really difficult spot because we were really close to the end of our runway, and we had to find a way out. And we saw from the data, because we follow, of course, the global investment data, we saw that none of the early companies in phase 1 or earlier were able to raise basically any money. So raising 20 million seemed quite impossible. That means because the GMP batch essentially is so expensive. So that sort of defines the price of the phase 1 study.
How do you mentally prepare when you're faced with that situation? The disappointment of thinking that you're nearly there, you're just about to move into clinic, your dream's going to become a reality. How do you personally dig deep and start to overcome that level of disappointment?
I just, I'm maybe a sort of person that I refuse to give up. I just, because we are living and breathing all the time, this company. So we really started thinking, and of course we knew that we have the discovery technology. We knew that there's a number of different genes that can be regulated by RNA activation. We decided to, let's do the same thing as our competitors. So let's start the out licensing business. So contacting pharmaceutical companies and trying to do co-development deals with them. So that would mean we would develop RNA activation drug to a certain point, which it would be out licensed to a company with some upfront payment, some milestones, and some royalty in the end. Our competitor, Therapeutics, which has been around roughly 15 years, that's in London, they made a little over 3 years ago massive deal with Eli Lilly, which is the largest pharma in the world. And they are developing RNA activations for 5 different indications, and they got 25 million upfront. Possible milestone payments are up to 1.2 billion, and then a royalty on top of that. So there is this market, and we just have to get big. We just have to convince big pharma that we really know this technology inside and out, and we can really do this. And so needs a little bit more proof of principle demonstration from our side. And
when you began to start looking for those partnerships, it's a very different skill set to science and discovery.
Oh yes, and all in all, I would say, having long academic career of 23 years and then jumping to a startup scene, you just have to learn so much new and everything. And so my tactic was always that I just keep a straight face. If I don't understand what the other person is talking about, I just nod and yes. And then afterwards, Google and find out what the hell he was talking
What they're
about.
talking
Yeah, so it's just you need a good poker face, and you have to be
Yeah.
fast learner and adapter. So
Yeah.
you have to adapt to situations. But I would say my father was a sales manager in Valmet, which is
Okay.
did his career there and was successful in his career. So I think I have the same characteristics as well. So
Yes.
even though deep down I'm a scientist, but at the same time I'm very capable business development person.
Yeah. Evidently, because you got yourself out of a huge sticky situation and managed to start generating revenue for the business.
Yeah, that is the current aim. Current
Yeah.
investor now, which joined this summer, is Nordic Science Investments, and we are now their portfolio company, and we are very
Yeah.
happy of that. So we get a sort of like rebirth situation, and therefore now we are putting a lot of effort on this platform business, and therefore we are currently in process of recruiting new CEO for the company, and that is going really, really well, the process at the moment. We are in the middle
Yeah.
of interviews.
Fantastic. It's very exciting. When you reflect back, what would you say are the biggest lessons that you've learned that have allowed you to achieve what you've achieved today?
Just biggest lessons. Um, Maybe just to never give up and trust
Yeah.
the data and trust your own instinct.
Yeah.
I really believe that we are still in very early years of molecular biology. For example, there's no such thing as junk RNA, which
Yeah.
was a concept 20 years ago, what some people used. But RNA is there for a reason, and we don't understand 95% at least. We don't understand what it does, but it is there for a reason. It would have been lost in
Yeah.
evolution long ago if those non-coding RNA transcripts wouldn't be important for the function of
Yeah.
life. And that is actually where the philosophy of RNatives comes from. We believe that the RNAs that we use, which are short non-coding RNAs, are the early form of life.
Yeah.
So that's what the RNatives name refers to, actually.
Yeah. Oh, that was fascinating. And like you said, you've been sort of 27 years in gene therapy as an expert. The discoveries have changed so much over that time. Do you have any predictions of what the future looks like for gene therapy?
Certainly, artificial intelligence and machine learning will change the game, and that's why we are also utilizing that to our software together with the AI partner. That will greatly help the sort of analysis of the non-coding RNAs, which I said that we really don't know majority of them what they are doing. So these guys, because there's so much data on genome browser and all the other data banks. It's just sitting there, and we have to find correct ways to use. Because currently, many things that me and my wife do when we do the discovery process, we do manually. But the reason why we want to integrate AI into our software is to speed up and to make it more accurate and predictive of the function. So I think that will be a biggest game changer, and that we already see. So basically, we are in a knowledge economy at the moment. So it's information that we are selling.
Yeah, absolutely. If you could go back and give your younger self, or to somebody who's thinking about moving from academia into industry or finishing their PhD, what one piece of advice would you give to them?
Yeah, let's say so you have to observe what is happening in academia and in industry all the time, because things are changing. My original plan was to go do a career only in academia and become a professor, but it's now changing, and for example, in Finland there will be more and more PhD students and PhDs in the future, and so try to pick the trends and work with the best ones and try to find and don't be afraid to move abroad and to live in different cultures, because you always learn. Basically, I need every skill that I've acquired in Finland and abroad. So in the development of this, and all the contacts, they are super important. So try to network as early as possible. Use LinkedIn and try to generate contacts and people who can sort of be your reference in the future. So that's super important.
Yeah, that sponsorship, that mentorship. And like you said, you can go off and drive a red Mustang and get a lovely suntan.
Yeah, so there are positive flip sides as well.
Yeah. So we've talked a little bit throughout our discussion around RNatives being in a really positive position, hiring the new CEO. Once that's complete, what will be the next step? How will your role change once the new CEO comes into play?
It will most likely be a remote job. To find that kind of talent in this very specific field in the biopharma, can't be found from Finland, and I understand that top talent might not be very eager to move to Eastern Finland in Kuopio, which is quite distant place, to be frank. So I think there will be more and more travel, and more business development. The CEO will need me also in the business development to be there to pitch and sell, because I can argue and litigate the case from the technical point of view very carefully. So so therefore he or she to be hired must be somebody I can work like hand in hand in the business development. And it might be that I have to move to US back again. I wouldn't mind though. I lived 3 years of my life already in the US, because I was as a teenager. My family lived in Atlanta, Georgia, for a couple of years. So as a teen, we sort of got affected by US culture and all of that. It sort of certainly changed me and made me more outgoing and more confident on talking. It's a different culture, and in US, I was surprised. Well, what I found it funny that if you remember the Snoopy and the Peanuts cartoons, there was always this lemonade kiosks that kids were selling lemonade for 5 cents or whatever. So
on their driveways,
and I saw that in San Diego when I was there. There were like 7-year-old kids, selling lemonade for $1, and I was okay. This is really entrepreneurial country, and people are encouraged on that. So I think that's why Finns are sort of reserved people, maybe would be maybe correct word. So for especially Nordics people, I would definitely recommend to travel and to live in different cultures. It really improves your person.
Sort of add to, I find people from Nordics are often very modest, which is quite the contradiction to the US. So it can widen that scope, and you end up maybe somewhere in the middle.
Yeah, exactly.
Well, it's really exciting times for you and RNative. It's wonderful to hear such a positive story. You've overcome so many huge hurdles. Not many companies would still be here today, and I think you need to take a massive pat on the back and huge credit for what you've achieved so far with the business.
Thank you very much for your kind words. Persistence and luck, I would say. There are sometimes small things Small
Yeah.
things can make a huge difference. And I tell you, this is a business development advice for similar startups. Don't go to the biggest events like Bio International or those that are massive with 25,000 people attending. You won't stand out. You won't get good meetings enough. Go to as small as possible business events. They are very effective, and so don't overlook them. They are usually quite cheap, even free to go, and still you might have the investor there who believes you and starts
Really in
powering
the
it.
niche. Yeah, I can
Yeah,
understand that. Yeah.
that's actually how we survived.
Was it? Okay. Fantastic advice then for your peers that will be listening.
Hopefully.
It's been an absolute pleasure to talk with you, Mikko, and to hear your career story. Thank you so much for sharing.
Thank you, Emma. It was a pleasure talking with you.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.